Good morning. Thank you for joining us. Today, we are hosting our second virtual seminar in North America focused on new advances in live cell imaging and analysis. My name is Victoria Yatsula. I am the Regional Business Manager for the Bioanalytics Division at Sartorius North America, responsible for cell analysis platforms. I will be moderating this session. Before we start, I wanted to let you know that we had some useful resources added for you in the handout section, so please feel free to download. And if you are interested in requesting a meeting from your local sales and technical experts, there is an option to do this during the webinar. You can simply follow the link in the chatter to request a meeting. If we can advance to the next slide, we selected several interesting topics for you today. We will start with the InQSight technology background, review techniques and methods for real-time analysis of cell health and function, subpopulation identification, looking at individual as well as adherent and non-adherent cells in mixed cultures, with examples of three-color analysis. Other topics will include metabolism assays, and we will conclude with organoids, review our new software and assays for organoid cultures and kinetic readouts. I would like to introduce our presenters. We have here Mark Metcalf. Mark manages a team of field application scientists in the South. And we also have a pleasure of having with us today Minnie Oliver. Minnie is a scientist in the group that developed the organoid software. Without further delays, I will pass it to Mark.
All right, thank you, Vika. I appreciate it. Thanks everyone for joining us on the call today. I wanted to, like Vika said, I wanted to talk a little bit about some of the new applications that we have to offer on the InQSight live cell analysis system. But first, I wanted to talk very briefly about what we're doing in the lab on a day-to-day basis. So this might be a very standard assay that you're doing in your labs where you're potentially doing a drug response. In this case, we're looking to see if a drug cocktail has any sort of synergistic effects, rather comparing that to the single drug. So it may take a time point at 12 hours, or again at 24 hours, or if we're disappointed with the response there, we may even go on out to 48 hours. So if we see a reading like this, we would assume naturally that this drug plateaus off and that the drug cocktail has no apparent effect above and beyond what the single drug was displaying for us. But what we might be missing is all of that information for those time points in between. And that's really one of the strengths of the InQSight platform, whether it be the SX1 platform, which we'll discuss here in a minute, all the way up through our SX5. We allow you the capability to kinetically monitor your cells whenever they're going through these changes and in their environment. So the InQSight sits completely inside that cell culture incubator. And then we're capable of imaging your cells in a variety of different microwell plates, flasks, dishes. But the real beauty of this system is that the cells are remaining where they are in the instrument. The optics are what's doing the movement to your cells. So you're not sloshing them around, trying to move them to a microscope or taking them out of that incubated condition. And one of the beauties of our scheduling software is that you can schedule different optics. You can schedule a different magnification to take images of your cells on different schedules with different fluorescence magnification or different fluorescence channels or those different magnifications. And any of our InQSight platforms are going to have a very simple workflow. So we provide you with some protocols, a suite of validated reagents that allow you to easily set up your cell culture experiments. Then once you have that assay set up and prepped, you can place it into the InQSight instrument completely inside that cell culture incubator. And then you can use the networked ability of the instrument to schedule your experiments or even view your images that have been collected, all from the comfort of your desktop outside of that lab environment. You can acquire those images as long as you'd like, in whatever channels you'd like to acquire those images and those fluorescences. And then once you have those images collected, the real beauty of this system comes in that you can apply analysis directly inside the IncuSight ecosystem so that you can quantify those cells and their changes whenever and wherever they're happening. So like I said, we have a family of these IncuSight instruments. So for those of you that still might be using the Zoom platform, we now have the SX1, the S3, and the SX5 instruments. So the SX1 is going to be somewhat similar to the Zoom platform in that we have a serial workflow. All of the vessels are going to be scanned on the same schedule with the same objective, with the same fluorescence channels. We still support over 600 cell culture vessels, but this is a value-oriented option that allows you to scan 2 microplates in parallel at the same time. All of those objectives are going to be installed on a turret so that you can easily switch between them whenever you're ready to clear that scan schedule and start a new set of experiments. The IncuSight S3 platform has really good multi-user support. It's got capacity for 6 microplates at a time in parallel, and it offers you a flexibility to accomplish several different experiment types. But for those of you that need the ultimate in flexibility, the SX5 is our newly introduced platform. It's the newest generation and has the highest capability. It has up to 5 different fluorescence channels, 3 of which can be installed in an optical module at once. We give you the capacity for 6 microplates, and again, 4x, 10x, and 20x objectives. So this is a very powerful piece of equipment so that you can perform a variety of different applications. The stock configuration, by the way, is green, orange, and near IR. And we have reagents to support multiple different applications in all of those fluorescence channels. So again, what we're going to be covering today are some of the real applications of this new hardware and some of our new software that allows us to analyze your cell culture experiments in new and exciting ways. So I'll show you first some apoptosis data. So this is one of the most common applications on the IncuSight platform. We're going to be plating cells in a plate. These just happen to be nuclide red labeled cells, and we've added in caspase 3.7 to sense when these cells are dying. So just like on any IncuSight instrument, we're going to image these over time. So here's data from 0 hours. Here's data from 30 hours into this experiment, and we can see much fewer cells that have been treated with this camptothecin at 1 micromolar, and a lot more cell death in that green channel. So rather than just looking at this in areas or the amount of the count or area of green or red fluorescence, we can now individually enumerate these cells, as you see in this aqua colored mask. But more importantly than that, we can tell you the amount of fluorescence on these channels, and we can graph it in new and exciting ways. So here's a dot plot where each of these dots is actually the intersection of the green intensity and a red intensity of a masked object. So we mask them in phase contrast, and then we plot their green and red intensity on this dot plot. So we can see how these subpopulations of cells go from just red or just green, where they're dead, to this intermediate sort of early apoptosis where they express both green and red fluorescences. We can plot the total number of cells that are in each one of these quadrants or a percentage of the population. Here you can see that with a high concentration of camptothecin, we have a fairly quick drop off in the amount of viable cells in this population. And we're doing this not only for just a single treatment group where it's plus or minus drug. Here we have a titration of a lot of these different drug concentrations, and we can tell which cells are in this early apoptotic state or which ones are viable and are just red. Here's that same data, but here we're showing it on non-adherent cells. So I've got a nice little video that shows how these populations shift from quadrant to quadrant over time. And we've also got this live graph over here showing you what percentage of the population is going to be viable, undergoing apoptosis, or is going to be in late apoptosis as indicated by this green group. And we're almost sort of backgating here so we can see which cells within this population are going to be falling into each one of these quadrants. Obviously, early on, a lot of them are going to be red because they're viable, and then we can see them shift to those populations as we go through time. And that's just one of the really nice applications using the cell-by-cell module. Another neat one is going to be cell cycle. So I'll play this video for you guys, and we can watch as these cells divide and lose their green fluorescent protein. Then when they switch over and start to replicate their DNA, they turn this reddish orange color, and then they end up back in the G2 phase right before mitosis. So we can watch these cells dividing. We can look at interesting phenomenon like multinucleation as well. And we can watch these across multiple divisions. So here we're showing you where those cells fall in each of these populations. And if we synchronize ourselves with something like thymidine, we can also watch how these cycling events take place and look at the cycling period of these cell populations. So it's a really valuable application that can use an existing sort of cell cycle sensing probe. We also have our own lentivirally transducible probe, where it's a single lentivirus and both green and red fluorescent proteins are expressed on the same plasmid. So you don't have to worry about constantly retransducing your cells, or maybe not getting an appropriate transduction of both of those plasmids. In our case, they're both on the same plasmid. Another unique application is going to be this neuronal activity application. So we have a calcium sensitive mRuby protein, and when calcium floods into these cells, we're going to see an increase in orange fluorescence. So here we're showing you a video that's a little bit sped up, and these neurons are actually forming a network over time. So this is the same well at day 4 and at day 12, and we can see at day 12 we have a much more concerted, much more highly correlated flashing of this neuronal network. So here is a view of the all objects trace. Here we look at all of the active cells, and we can see all of them here at day 4, fairly low burst intensity, fairly low correlation. But as we move on out to day 12, we can see these really nice bursting events here, where we can see 7 bursts that occur in this approximately 3-minute time period with very, very high correlations. And we can graph these for you over time. So again, we can see increasing correlation here in the black graph, and then we see an initial peak in the burst rate, and as they become very tightly correlated, that burst rate declines a little bit as all of these cells are bursting with this extreme fluorescence intensity. Another one of our very nice, sort of recently introduced applications is going to be not only the spheroid application, but the capability to do this for multi-spheroids. So for those of you that might be on the Zoom platform, you would have to use a fluorescently labeled cell line to get good masking. Or even if you don't have the spheroid module on your S3 platform. If you get the spheroid module, we actually take images in bright field so that you can perform better masking without a fluorescence label. So I'll play these videos. On the left-hand side, we have normal 231 cells that are on top of a Matrigel layer, and we're imaging in this multi-spheroid mode. On the right-hand side, we've treated these with camptothecin. So a lot of these cells are dead, but it's not quite apparent here. But if we can add in a fluorescent marker, we can look at how these cells change their fluorescence intensity over time. So these cells are going to be dying, and when they die, they lose that fluorescent protein expression in their nuclei, and they become, on average, lower fluorescence intensity within these identified objects. So on the next slide here, I'll show you what some of this data looks like when we graph it. So here on this half of the slide, we're looking at cells where we're just looking at the bright field metrics. So we see an increase in those bright field area for the DMSO treated control. And then we see a slight increase, but it sort of tails off with the camptothecin treated group. Now the difference is more pronounced here when we have a red object fluorescence in here. So we can see a reduction in the amount of red fluorescence intensity over time. But keep in mind, we saw on the last slide here, we're only looking at the red fluorescence intensity inside these boundaries that we've identified in the bright field channel already. So we're ignoring any background fluorescence that may have been coming from the Matrigel or from the media. So it's a really nice technique when we can just limit it to that red object area within a bright field boundary. Another very exciting application that's probably my personal favorite of the ones that we've introduced recently is going to be live cell immunocytochemistry. So here we have some A549s that are constitutively labeled in red, and we have some T cells where we've actually used this reagent to label CD8 on the cell surfaces. So when we allow these cells to interact, you can see that these T cells are forming a synapse here with these A549 target cells. And that CD8 expression is very concentrated at that synapse that's formed. So in this next slide, I'll show you just that one interaction. And we can quantify this for you as an overlap area and give you these exciting videos to prove that we actually have specific organization and specific interaction between our target cells and your immune cells in this case. So here's another example of an immune cell killing experiment. Here we've labeled our T cells with an anti-CD45. So these are just PBMC that we've activated, and we're looking for CD45. And we have a 3-color experiment here, this sort of cornflower blue color on the nuclei. That's how we pseudo color the near IR channel. It's just a little bit easier to see instead of a very deep red. But here you can see we're looking for that CD45 marker. But if we are looking at an isotype control, we do not have any of that green fluorescence expression. So again, we can show this in a dot plot. We can show it in a histogram. So here's that green marker. The green group here are going to be our CD45 positive cells in the group that has the Fab448. This yellow group is our isotype control. So we see this very nice shift here when we've applied that Fab448 labeled CD45. So, and this live cell immunocytochemistry reagent is useful in a vast variety of applications. As long as you've got a cell surface marker expressed on your cells and you have a good antibody toward it, you can watch as that marker expression goes up or goes down over a time course in your live cell experiments. So just to sort of summarize how this would work and sort of to highlight our 3-color applications, I'm going to show you how to actually go through and analyze some data very quickly using the immune cell killing experiment. So we'll start here and play this video for you guys. So we'll open up an experiment that we've already acquired. And again, that's 3-color data. We have CD8 positive T cells in the green channel, our Ramos, our nuclei orange, and a Nexin-5 near IR is going to be our cell death indicator. So we can create an analysis definition for this. Here we're going to use the basic analyzer first, and then I'll show you how non-adherent cell by cell works. So for the interest of this, we're going to exclude the phase contrast channel, and we're going to look at overlap for all of these fluorescence channels as well. So I'll select all of these so they're analyzed. Now, since we have 3 colors, there's way more overlap channels, obviously. And we're going to select a set of images that we can use to preview and refine our analysis definition. So here I'm choosing a activated PBMC group versus a PBMC group without activation, both at an early time point and at a later time point, so I can see these changes dynamically. All right, so the first thing I'll do is I'll click on preview so we can see what the baseline analysis definition settings look like. And it applies these colored masks. So first I'll look at the CD8 positive T cells, and I think we're not getting as much area as we should. So what I can do is I can look at the relative fluorescence of these cells. I'll reduce the background radius there, which will be a little bit more stringent background subtraction, and set a little bit lower threshold and turn our edge split off so that we get more of that green area recognized by our masks. And that looks substantially better to me. I'm pretty happy with that in the green channel. So I'll look at orange. Again, those are our Ramos nuclei orange cells. So I'll again increase that stringency of the background subtraction, increase my threshold just a little bit so I don't get much background, and I'll leave that edge split on since it's nuclei. I'll change the background subtraction there on the near IR channel, turn that edge split off, and the threshold should be good around 2 near IR corrected units there. So again, here's this preview. It's an early on time point where we have activation. I can toggle these channels on or off to make sure that I'm quantifying near IR correctly, or that I'm quantifying my target cell nuclei correctly here with this mask, or that I'm quantifying my CD8 positive T cells correctly in this green channel. So I'll check out here where we have no activation of our PBMC. We should have much fewer cells in green there. Here we're looking at our isotype control. Or actually, this is a later on time period, excuse me, of our activated PBMC. And I think these masks look satisfactory. So I can apply this analysis definition to all those time points and all those wells. And then you have to give it a unique name here so that you can keep track of this analysis definition. Like I said, the name has to be unique, so we'll change this very slightly. And then we'll apply this analysis definition. Okay, so that's how we would set this up for an immune cell killing experiment in 3 colors. I've actually already run this analysis definition, so I will minimize this out and we will take a look at what the data analysis looks like. So I'll open up that analysis definition here. And then very quickly, I'll toggle these channels on and off to make sure that our analysis looks robust. And in fact, it looks pretty good there. So now what I can do is I can go ahead and graph this data. So first of all, we'll look at the area in the green channel. Again, this is going to be CD8 positive T cells. Over here on the left-hand side, we have the highly activated cell population versus the isotype control. And we're also looking at the CD8 there. In the orange channel, these that have activated PBMCs, our targets are not proliferating very much, as opposed to those where we don't have activated effector cells. They're proliferating very well over the course of this experiment. Again, in near IR, we're going to be looking at cell death. So we have a higher amount of cell death in those populations where we activated the PBMC, and almost none down here where the PBMC were not activated. So this is a very good explanation of how this can work in the basic analyzer. But again, one of the real strengths of this is that you can analyze these 3 color assays using cell by cell mode. So here we've already identified which cells we've already identified these objects in phase contrast, and they're circled in yellow. But now what we can do is actually go up here and set what's called a classification definition. And that allows us to plot these events, just like I said, in a dot plot, and then segment them based upon their fluorescence intensities. So I'll select here on the plate map the isotype control, those that are not activated, that don't have activated PBMC, and we'll look in the orange channel for our nuclei of our targets, and we'll look at cell health reagent there in the near IR channel. So we can see a lot of our targets here are in this population to the right, and our effectors are on the left. Just quickly set the near IR threshold there for what I want to call as positive death. Notice I'm looking here where I don't expect there to be much death, where I don't expect there to be that much activation, and I'm setting my thresholds in this group. So then you'll see me look at a different time point here, where I'm trying to ensure that there's, see what level of death we have. If you look over here on the right-hand side, it's only about 5% of those cells are going to be positive for a Nexin 5 near IR, which is about what we would expect. So I'll go back and I'll look at some of these wells where I actually have activated PBMC. And again, that's my isotype control versus my CD8 positive cells. So if we look in this case, we have a little bit more death here just in that early time point, but as we go over time, look at my targets over here. A lot of those targets are shifted upward, so we have a lot of those cells that are dying. And even out at 1 day, we almost have none of our cells remaining, none of our target cells remaining that are not dying, particularly as we go through time. So I can apply this classification. You saw me just move those thresholds around. I'll apply it to all of those groups, and then I can give it an analysis definition name once again, and then we can see how those analysis modes shake out once we have applied these. So rather than going all the way through this video and looking at these graphs, I think that classification was a pretty good way to sort of summarize the way that this module works. So I'm going to proceed on to the next slide here. And I will tell you a little bit more about another application called ATP metabolism. So briefly, this is a lentiviral-based reagent where we have a sort of a dual excitation, single emission setup that allows us to compare the ratio of fluorescence when ATP is bound, where we would have high fluorescence when we excite at 485, get that energy transfer, and then read out in orange. Versus when ATP is not bound, we would have very little energy transfer from this green fluorescent protein to the orange, and therefore have a very low readout in the orange channel when exciting with this blue LED. So what we can do is transduce a target cell line and have a constitutively expressing ATP sensing line. Over here on the left-hand side, we have our cyto-ATP lentivirus, and over here, we have our lentivirus that has not been exposed to, well, it's actually a null lentivirus. It doesn't have this sensor in it. In this video here, we've added in some 2DG, so we should see a reduction in that ratio as indicated by lower fluorescence in the, or lower mask intensity in the yellow channel here. But over time, the ATP builds back up in these cells, and you see these cells getting brighter and brighter yellow, indicating the ratio of ATP to the amount of fluorescence in the null lentivirus. So again, you can see a dose-dependent reduction in the amount of ATP present in these cells whenever we've treated it with various different concentrations of 2-deoxyglucose. One of the really, really nice things about this application that I think makes it stand out over pretty much any other method of measuring ATP metabolism, not only is this a kinetic method, but since you can make a constitutively expressing target cell line, and we're only looking at those cells that have been effectively transduced with this reagent, is you can actually perform this in a co-culture. So, you know, we may be interested in the way that our cells utilize energy, but for tumor cells that are doing this in a actual living human being or in an animal model are going to be surrounded by a lot of fibroblast sort of supporting cells. So when we do this in co-culture, we can look for subtle differences between the way that different cell types interact with our constitutively expressing ATP sensing cell lines. I also wanted to point out here that if you guys are using any software that's older than 2019B on the S3 platform, you won't have IC50 data as a plottable readout. But if you're on any newer software than that, this is a feature that's already embedded into that software in the basic analysis mode. You can just quickly click on concentration response, and you can generate IC50 curves for your samples. And you can do it over every single time point that you've performed these readings. All right, so with that, I wanna go ahead and turn it over to Minnie so she can tell you a little bit about the differences between the spheroid module I already discussed and this organoid QC. So take it away, Minnie.
Thanks, Mark. So as Marcus mentioned, I will spend the next few minutes just going over the IncuSite's new label-free organoid QC imaging and analysis software module. And this module really just focuses on organoid 3D model workflows where organoids are embedded in Matrigel domes. So the next slide here, I'll just give a very top level overview of some of the key advantages of this approach. You know, one of the first things that really stands out about this approach is that it allows you to automatically locate organoids individually embedded across multiple planes within a Matrigel dome, and it allows you to quantify organoid differentiation and maturation. You can also utilize this module to characterize organoid formation with some integrated real-time kinetics. And you can measure things such as size and count really rapidly, and this really enables unbiased assessment of organoid maturation and provides great insight, particularly when it comes to determining optimal passaging periods or culture extensions with regards to organoids. And this solution, alongside a fully validated protocol, which I'll just go through briefly on the next slide, enables easy generation of reproducible organoid formation analysis in 24 and 48 well plates. Now here we have the schematic of the protocol, which just really gives, as I said, a very top level overview of how embedded organoid cultures can be prepared in either 24 or 48 well plates. And this is essential for optimal imaging and subsequent analysis using the IncuSite. So briefly, your cells of interest or organoids of interest are harvested according to the model-specific guidelines and resuspended in Matrigel. The Matrigel solution containing your organoid fragments or cells can then be pipetted into the center of a 24 or 48 well plate. The plate can then be placed at 37 degrees to polymerize the Matrigel. And following polymerization, the plate can be removed from the incubator and the addition of model-specific growth media can occur. And finally, the plate can then be placed in an IncuSite to monitor organoid formation and growth. So on the next slide here, what we can see is that using this proprietary novel imaging acquisition approach, the IncuSite is able to consistently acquire bright field images of organoids embedded within Matrigel domes. Now shown here are bright field time-lapse videos monitoring the formation and growth of intestinal, pancreatic, and hepatic organoids using this organoid software module in 24 well plates. Now, one of the first things that really stands out here about the software is that it has great ability to visualize the entire dome. And within that, you can see you can consistently image individual objects within that dome over time. And what you can see, and you will agree by looking at these videos, is that it also demonstrates that the software is able to image distinct organoid morphological features, such as the intestinal organoids seen here on the far left in the video. We can see that this particular cell type has a very unique morphology in comparison to the other two cell types. It exhibits this budding around the lumen as they mature, and this is characteristic of this particular organoid cell type. And you can see that the software does a really good job at imaging this morphology, as well as the more rounded phenotypes of the pancreatic and hepatic organoids, which appear much larger over time. So we can then go on, once we've acquired these images, to quantify this organoid differentiation and growth over time. And shown here are some example time-lapse videos of intestinal and pancreatic organoids taken 2 days post-seeding in 24 well plates. And we can see here the yellow outline mask around each of these objects, and this represents the bright field segmentation, which enables a label-free quantification of changes in organoid size over time. And we can see from the corresponding time courses on the far right here that the software is able to track changes in size and area and as a total population within the dome. And we can see that this is really cell type specific when we compare the two cell types shown here in this example. So now defining the optimal culture conditions, such as cell number or passage frequency, is essential for maintaining or expanding healthy organoid cultures. Organoid passage frequency is often driven by various indicators of maturation, and that's all cell type dependent. But for example, maturation can be instigated, or it can be tracked by changes in size, the accumulation of debris within a lumen, for example, or even the budding phenotype I described earlier with the intestinal organoids. So to exemplify the use of this software module to objectively define optimal culture conditions and track some of these indicators of maturation, we performed a seed and density titration study with hepatic organoids, which is what you see here on this slide. So hepatic organoids are typically ready for passaging when the majority of organoids have reached their maximum size and have not yet collapsed. So if we just take a few minutes to just look at the images that we can see at the top here, these representative bright field images illustrate that 2 days post-seeding, so that's the first image we can see there on the top left, cultures are not yet ready for passaging, as the majority of these objects within this well, you can see they're quite small. They're about less than 100 microns in diameter, and they exhibit clear lumens. Now, by day 4, which is the image in the middle there, we can see that most of these organoids have increased in size, and you can see that most of them appear to have reached maximal size. They exhibit clear lumens, and they all exhibit a rounded morphology and have not yet collapsed. Now, when we look further in time and we look at the day 6 images on the far right there, we can see quite clearly that from the images that the health of this culture has deteriorated as the majority of organoids have collapsed and darkened. So now some of these changes that I've just described, such as the changes in size and morphology, can easily be kinetically quantified using integrated metrics associated with this organoid software module. We can see here, if we focus on the first graph on the bottom left here for the total area, we can quantify the total area, which enables us to rapidly assess organoid growth and size over time. And looking at this plot here, we can see that the total area, it really demonstrates that organoids seeded at the highest density, which corresponds with the images that are shown above, they exhibit rapid growth, reaching maximal size within 96 hours. And again, that lines up quite nicely with what we saw, or what I described in the images, where at day 4, we can see that these reached maximal size. And when we look at that time course of the 1 in 10 of the total area, we can see at 96 hours, if they begin to plateau, and you see this reduction, this slight decline in the total area. And it's clear to see from the time course that the organoid growth rate and size is directly proportional to seeding density. So looking at data like this can help you determine what your optimal density is for your culture, and for what passage period is optimal as well for your culture. And note that while the optimal growth is obtained within 96 hours at the highest density, we see this continued growth in organoids at lower densities, where we see growth beyond 144 hours. So if we focus now on the graph in the middle, we can see a decline in the eccentricity, especially at the highest density, again, if we focus on that, within 48 hours, as the organoids formed and became more rounded over time. So we can track this change over time, which is quite nice. Again, as I said, you can use these morphology and size metrics to really delve deeper into what your cultures are doing. So the optimal period for passage in this culture really occurred between days 4 and 5, when most of the organoids within the dome had reached their maximal size, they exhibited a rounded morphology and had not collapsed. Now the time course on the far right, tracking changes in brightness over time, shows a marked increase in organoid darkness as collapsed organoids darkened over time. And this is a really nice new metric that's specific to this module that really gives you extra insight in what's happening within your culture. And we can see that beyond the 4 days, as I described, the culture is beyond its optimal passage period. And again, you can see that quite nicely, that this is seeding density dependent. And this is just really just to give you an example to show you how this tool, how using this tool and these imaging and alongside imaging and quantification, you can really minimize the need for extensive training when it comes to organoid cultures, because these are quite novel. So sometimes they require quite a lot of training, and it really ensures consistent, unbiased, operator independent regimes are employed. So now I will just spend some time walking you through the software and demonstrating assay setup and analysis using this module. So as you can see here, we have the acquisition window, which is used to schedule and acquire images. So the first thing we do is just select the basically trying to add a vessel, and here we have two options. We can choose to schedule it on repeat scanning mode, or we can scan it once, but we'll go with this first option for this demo. And here we can then create a new vessel. So you've got various options, but we'll just create the first one. So now here we have the scan type page, and which lists the different ways in which the vessel can be scanned. So selection is based on the assay of interest and the application of interest. And here in this instance, we're interested in organoids, and it's important to select the correct one, as these are associated with specific acquisition and analysis software. So as I said, so selecting the correct mode is vital. So here we're going to click the organoid, and here on the next wizard, we just have the default settings when it comes to the image channels and the objective for this particular module, and they're grayed out as these are default. We then can select the plate of interest, and as I mentioned, this module supports the 24 and 48 well plates, so you can select whichever plate of interest that you have. We then on the next part can select what position if you're familiar with the IncuSite, you know that you can have up to 6 plates running at the same time, and the same is true for this particular module. We then can select all the wells that we are interested in imaging, and the rest of the wizard is quite straightforward. Here we just name the vessel what we want to name it, whatever the cell type is. For instance, in this example, it's hepatic. You can also add a plate map if you've got different conditions in your plate. And then the next bit here, you just can create a scan schedule. So we scan these every 6 hours. And this gives you a nice summary of everything that you've scheduled. So you can just double check everything before you add to the schedule. So once the plate has been scheduled, it will then scan. We then can go to view to visualize what we've scanned. So to view the scanned images, we just double click the scanned vessel, and the vessel view or deep view window is then displayed as we can see here. And this is just an example of a seeding density titration hepatic organoid assay plate, which allows us to visualize all the scanned wells within our plate. So what we can then do as well is scroll through time. So immediately open a job, a vessel, you get the most recent image acquired, but you can scroll through time to really see how your biology changes over time. And this again is going to be really vital for some of the next steps I will show when you're creating a new analysis definition. It's imperative to get representative images from all the different time points and all the different conditions. So here we'll go ahead and create a new definition, even though you've got other various options if you've created one before. And again, here we just have the 2 channels that are supported for this module. So as I mentioned, and Mark mentioned earlier as well, selecting images with key conditions is important. I'm just going to select a couple of wells here at different time points just to exemplify the use of this analysis module. So I'm just selecting some at different densities, just so we can see how the software is able to segment high density and low density wells. So shown here on the left are all the parameters are required to define an optimal segmentation using this module. And a lot of these people will be familiar with if they've used an IncuSite before, but there's some specific parameters for this module. And the first thing I always do is preview the current settings, because most of the time this is a great place to start. So you can then interrogate and see how well the parameters are doing prior to changing any of the default settings. So I'm just going to increase the width here so we can have a closer look at what exactly the segmentation mask is doing. And it's doing a reasonable job. It's segmenting all my organoids of interest, but we can see quite clearly that it's segmenting some objects that aren't organoids or that aren't quite mature enough. And here we can see that there's 2 organoids that are being masked as one, so we can use the edge split slider to try and separate those as well. So by moving it towards round, since this cell type has a rounded morphology, we can see that that has a really nice separation now of the two objects there. And you can zoom out and check if there's anything else that you want to refine. And I think I'm happy with that in terms of segmenting all the objects. But as I said, we can see some of these really small non-organoid or immature organoids being segmented. So I want to eliminate those, and I'm going to apply here an area filter. And so to just do that, we just hover over the objects that we want to eliminate and see what size they are. And we can just add a minimum area there to exclude them. So if I preview this, that should hopefully get rid of that. And it does quite nicely. But you know, my eye is drawn there to the bottom of this dome here, where you can see something that doesn't quite look like an organoid. So you can zoom in to inspect exactly what this object is. And I find toggling on and off of the segmentation mask really helps in identifying what object is being masked. And here I'm going to apply an eccentricity filter to try and eliminate that, as that wasn't able to get rid of that using the area filter. So I'm just going to preview that. And so once you're happy with a segmentation of a particular well, you can then apply it to all your images in your image collection, which is what I'm going to do here. And you can see it's doing a reasonable job of this well. So I'm going to move to the next one. And you can see here that it's capturing some of this debris around the edge of the dome. So I'm going to move the background to cell slider just so that it sees less of some of this. And that has cleared it up quite nicely. And again, any changes that you make sure that you apply it to your entire image collection to make sure that it hasn't affected it. And here we're looking at a slightly more dense well, and I think the software again is doing a great job of segmenting all my objects of interest. So once you're happy with your defined analysis job, you can then run it across your entire plate. So again, similar to what Mark showed, you can select the time points of interest, or if it's still scanning, you can select analyze future scans, select all your wells of interest, give it a unique name for this particular analysis job, and then you can click next and have it run on your assay plate. So I'm just going to change that to make it unique. And again, here you just have a nice summary. So once your job is run, you can then open up your analysis definition. And one thing I always like to do is just to ensure, the first thing I do is put the mask on, just to ensure that it's segmenting appropriately. And I would expect it to, as I selected objects or wells, from various conditions across this plate. So that's one of the key things of doing that, is to then be able to visualize that your mask is appropriate across your different conditions and through time. So you can see here by just scrolling at an earlier time point, we can see that it's again segmenting everything. We can then go on to graph the metrics. And these metrics shown here are specific to the organoid module. Some of these are already showed on the previous slide, but we can quite nicely see by just plotting this entire plate, which is a seed and density titration. We can see the seed and density dependent count for this particular metric. And if we look at the area as well, I can plot that just to show you again that we have a nice titration across the different seed and densities. You can also visualize this as a microplate view, if you will, but I won't dwell too much into that because some of the data has already been shown using some different modules, but the principle is the same. So I will just hand over now back to Vika, who will just give a brief summary on everything that we've talked through today. Thank you so much.
Thank you, Minnie and Mark, for the very insightful presentation. Hope everyone enjoyed it and found it useful. We would love to hear your feedback on this session, as well as other topics for the upcoming events that you are interested in learning about. And in summary, so the examples of data that we shared today were generated with the cell-by-cell InQSight spheroid and the new organoid software modules. We also reviewed data examples for neuronal activity and ATP metabolism applications. Of course, there is so much more. We did not cover chemotaxis, scratch wound, or neurotrack applications, but these are also available. If you are interested in trying any of these modules, just reply to the original invitation that you received for this webinar from your local sales specialist, and we will be happy to work with you to initiate the trial period. We also to remind that the software that we featured can only be run on the InQsight platforms, SX1, S3, SX5, our current instrument models. If you are interested in scheduling a new instrument demo, please reach out. Depending on your project biology and throughput requirements, we would be happy to demonstrate capabilities of any one of the selected instruments in your lab using your cells and your protocols. We also have a wide range of reagents available, and these reagents are really safe to use with cells. These are non-protruding, specifically optimized for the applications that we featured, and of course designed for long-term live cell imaging and analysis. You can visit our eShop at ascentbioscience.com. And if you want to take advantage of our current end of year promotions, just reach out to your local sales representative. And we are actually ready to take some questions. We do have quite a few questions that came in. The first question is for Mark. Can I do cell-by-cell analysis on mixed populations of non-adherent cells?
Yes, actually, that's a that's a great question. So if you recall back on some of the data that I presented to you, we were looking at immune cell killing. So we actually had a group of PBMC that we had with that CD8 label, and we also had some Ramos cells that were labeled there. So those happened to be an adherent cell and a non-adherent cell. We did that with the basic analyzer, but we could certainly do cell-by-cell analysis on non-adherent cells too. That's one of our very common applications with maybe B cell targets and T cell effectors. And we can enumerate all of those cells there in those non-adherent mixed populations and then give you information about fluorescent markers on those cells.
Thank you, Mark. Another question for you. Does the ATP module require the use of special plates?
No, and actually that's one of the really nice benefits of it too. Like I said, it's a module that allows you to make these constitutively expressing ATP sensing cell lines. But since we're doing this on that InQsight platform, it's compatible with a large variety of tissue cultureware, whether that be plastic bottom dishes or glass bottom dishes. So no specific plate is required.
Great. We have quite a few questions that came in on organoids. First question, can I track individual objects in a well over time?
So currently this is not possible using this module. As I said, this is a population analysis module for QC purposes. However, if somebody is interested in tracking individual objects, as you know, with the InQsight, you have the ability to export your images, and use of third-party software could possibly be used to track these objects individually. But within the InQsight, we provide population analysis, so the total area within a particular dome or well.
Thanks, Minnie. One more question for you. Can I image fluorescently labeled organoids, and can I perform 96 well embedded assay using this module?
So fluorescent imaging is not supported for this module. Only label-free quantification is available. And as I mentioned earlier, this organoid QC module enables real-time kinetic visualization and quantification of organoids embedded in matrigel domes in 48 and 24 well plates only. So downstream 96 well applications are currently not supported using this module.
Thanks. So Oh, this we also have another question that came from an existing user. I am already doing non-adherent immune cell killing assays with MAIA-3, but I don't have the cell-by-cell module. Can I go back and reanalyze my experiments to get dot plots?
I'll take that one, Minnie. So if you are doing these already with 20x magnification, and if they're on these non-adherent cell by cell, so both of your cell populations are non-adherent, I don't see any reason why we couldn't go back and analyze those using the non-adherent cell by cell mode. So again, it would have to be at 20x because we need that very high resolution to accurately segment these cells into their individual outlines. But it should be doable with those non-adherent cells. You would just have to activate that module so that you could then go back and reanalyze that previously collected data.
Thank you, Mark. Minnie, one more question for you. Can I use an alternative extracellular matrix to Matrigel for organoids?
So we have validated the protocol using only Matrigel. However, we have previously used ECM such as BME for specific organoid cultures, and again, that's the use of Matrigel was specific to the organoids that we utilized. I think what we would recommend is for somebody wishing to use an alternative ECM is to perform optimization, whether that's for concentration, volumes, just to ensure that it's applicable or they're able to use our software using that alternative ECM.
Thank you. So we can take one more question, and this will be another one for you, Minnie. Can I spot multiple domes in a single well?
So spotting a single dome is probably what's recommended or optimal for this using this software module, because it ensures that you image your entire dome. So by adding more than one dome within a well, it reduces the chance that you'll be able to visualize your entire dome. So we wouldn't recommend spotting more than one dome in each of the wells, and that goes for both the 48 and the 24 well plate.
Thanks. So this was the last question that we can take now, but we will review all questions that came in individually and follow up with you. And with this, I want to thank our speakers. This concludes our presentation. Thank you for joining us today. We really appreciate your time, everyone.